Good morning, everybody. My name is Kirill. I'm a postdoctoral fellow in Dr. King's lab, whom you lovingly know as Caddy. It's been great to hear all the talks so far, especially the one before Caddy's talk. I think that lays really nice foundation for this bit, too. Okay, so my presentation is titled GABAergic Neurotransmission of GAT1 S295L variant. So when we think of a GABAergic synapse, GABA being the primary inhibitory neurotransmitter in CNS, GABA is released at the synapse where it activates GABA A receptors on the postsynaptic neuron that will cause synaptic inhibitory current that's pretty acute and will quickly decay as GABA is taken up and diffuses out of the synapse. And also there are tonic inhibitory currents that are mediated by extrasynaptic GABA receptors, and these are activated by the spillover GABA and the ambient GABA that's present outside of the synapse. And so transporters, both GAT1 and GAT3, are pretty important in modulating both of these signals because they determine how much GABA there is outside a cell and how quickly it decays at the synapse, perhaps. GAT1 is the primary transporter, is expressed in most brain regions and is expressed by GABAergic neurons. GAT3 is only expressed in certain brain regions mainly, like thalamus, and it's primarily astrocytic. There are multiple mutant variants in SLC6A1, which is the gene that codes for GAT1, that are associated with neurologic and developmental disorders. And today I'm going to focus on the S295L, Maxwell's mutation. So Maxwell has childhood absence epilepsy, neurodevelopmental delay. And the big question that I had from my work with Caddy was how is GABAergic neurotransmission changed in the developmental and epileptic encephalopathy associated with the SL mutation. And to study this, I've used the knock-in mouse model that we've already talked about that was done using CRISPR. This mouse model exhibits absence seizures, as has been shown in our lab and also shown by the group that presented before us. And also, as Yulia mentioned, cortex and thalamus are two critical regions in absence epilepsy. So when I'm going to be looking at synaptic changes, I'm going to be focusing on those two regions. So what are the pathologic changes in GABAergic neurotransmission with DSL, developmental epileptic encephalopathy? As Caddy mentioned, we know that GAT1 expression at the surface level and total brain expression is reduced in these cell mice. And also in synaptosomes and in HEX cells, we've noticed, in HEX cells, we've noticed when we express the SL variant of GAT1, there's reduced GABA uptake. And then in synaptosomes from the SL mice, there's also reduced GABA uptake. So this would lead us to hypothesize certain things that might be changed here at the synapse. So we know that there's decreased GAT1 expression and GABA uptake. So there's less GAT1 and the uptake is decreased. So what could this lead to? Lead to possibly an increased amount of GABA and longer decay. So perhaps these synaptic events would last for a longer period of time. Perhaps there would be an increase in the tonic current because there would be more GABA outside of the synapse. And perhaps there would be less recycling of GABA as it's not taken up. So perhaps a GABAergic neuron could run out of GABA faster. So I tried to test these three different aspects. So first I consulted the GAT1 knockout experiments. So one very prominent finding in GAT1 knockout experiments is an increased tonic current in multiple brain regions. It's been found in hippocampus, it's been found in thalamus, and been found in cortex, sole and cortex. This is a pretty small amplitude current that was found in the GAT1 knockout. So I looked for tonic current as well. I started by looking in layer 6 cortex and pyramidal neurons, and I compared homozygote SL mice with wild types. And I actually did not, I looked at 2 to 3 months old, and I didn't find a difference in the tonic current here. And this is also very, very small, like it's almost zero basically. 4 picons is a very small current. I looked at an older age because at that time, based on microdialysis experiments and some other things, we thought there would be an increased pathology in an older age. And the ends are smaller at this point, but I didn't see much of a tonic current in the heterozygotes or in the homozygotes here. So this, of course, is under the conditions that I used. Possibly you could see one under different conditions. I also looked in thalamus, and here I only had heterozygotes available. Thalamus is known to have a pronounced tonic current in the wild type, and as you can see, I observed it here, so 30 picoamps on average. And then the cell heterozygote had a very similar tonic current, so I didn't see actually a change in the tonic current. And then the other thing I looked at were changes in synaptic inhibition. So first, again, I consulted the literature in the GAT1 knockout. And when you look at miniature and spontaneous inhibitory synaptic currents, in terms of kinetics, these publications, they didn't show any differences. The only difference they saw was in frequency of miniature iPSCs in hippocampus, where the frequency was reduced of these events. But in the Braginel paper in 2008, when they listed large evoked events, then they did see a very pronounced difference in decay between the GAT1 knockout and the wild type. So I set out to do this experiment in the cell mice and see if I could see a similar phenotype and difference. So at first, I did it the same way that that group did. So I patched a neuron in layers 2, 3, and I would stimulate layer 4. And so what you see here is basically for each neuron, I would take an average trace, and then I would average the traces for each genotype. And here they're normalized and superimposed. And so you can see that compared to, in blue, a wild type evoked iPSC, you kind of have this like stepwise increase in this lingering current and return to baseline from the heterozygote and then to the homozygote cell. And you can also see when you calculate area under the curve, you see this kind of stepwise increase with the difference between the wild type and the homozygote being statistically significant. But I felt like a lot of the responses I was getting for these neurons were a bit variable. So I was looking at other publications on the subject that'll give me an idea of how to test this. And looked at this one study where they didn't use a knockout, but they used No711, which is a GAT1 blocker that was previously mentioned. So compared to normal evoked event, when you compare that to evoked event with No711, you get greatly increased amplitude. And actually what they also did in this study is they would do a train of evoked events, like you see here on the right, and they would actually see a decreased amplitude when you block GAT1, possibly because you would be depleting GABA, at least I thought. So I thought that if I try this kind of an experiment, I might be able to address two of the questions that I had. When I did this, so I would patch layer 2, 3 neuron, stimulate layer 1, and I did a lot more cells for this study. I found a very consistent difference in decay between the wild type in blue and then the heterozygote in red. As you can see, the amplitude of these evoked events, I didn't see a change in, but I did see a change in the decay time and in the tau, which is the decay constant. Then when I added a GAT1 blocker, and I used tiagabine, if you look at the wild type, what I saw also, compared to just the wild type response in light blue, in dark blue you can see what happens when you add tiagabine, much longer decay. Here in the bar graphs, I'm showing the percent change, percent increase for these different metrics. So you can see the amplitude didn't really increase when I added tiagabine, but then the decay really did, and it was very statistically significant, over 100% increase in the decay time and decay constant as well. When I added tiagabine to the cell heterozygote mice, these iPSCs, they also increased in decay. This was a statistically significant increase of about 40%, but it was a lot less than the wild type, and that difference was also statistically significant. And same for the decay constant. So I thought this was a very interesting finding. And here it is all superimposed, so you can see here how a normal iPSC in a wild type is pretty acute. There's increased decay in the cell, and if you add tagabine either to the wild type or to the cell, you take it out even further. And then I looked at a train of iPSCs. I actually ended up spacing them out a bit more in that original paper that I mentioned. And my idea here was to look at changes in amplitude of iPSCs within a train to see if there'd be a difference between the wild type and the cell heterozygote. So first, I looked at the wild type and a wild type when I block GAT1 with tagabine. And basically, the second and the third iPSCs would always be smaller than the first. And here they are plotted second and the third as a percentage of the first. But what I actually found is when I added tagabine, instead of decreasing, the second iPSC and the third iPSCs would increase relative to how they would be without tagabine. So that's what you can see here between the light blue and the dark blue, light blue and dark blue. And then the cell mice, those amplitudes were kind of in between. Like, this was not a statistically significant difference, but you, I think, still can appreciate this kind of stepwise increase. So I don't think, based on this observation, I don't think there is a deficit in releasable GABA, but there might be a better way to test this. And here are the superimposed responses. But what you absolutely can see, though, is, again, this very clear difference in decay where each one of these events in the wild type, when you add tagabine, has an increased decay. If you compare wild type to the cell heterozygotes, there's an increase in decay. And if you add tagabine to the cell heterozygotes, you also get an increase in decay, but this increase is not as significant as it would be in the wild type. And here you can see all these responses superimposed. So after I talked to some of my colleagues about these findings, we were saying that if we're seeing these differences in evoked events, then often an evoked event is a summation of many synaptic responses when you stimulate multiple inputs to a neuron. So you would expect to see some of these kinetic differences also on the synaptic level, so in spontaneous events. So that's the next thing I did. In these exact same cells, actually, I looked at spontaneous iPSCs, and here you can see two average traces and an average response from each of these cells. If you superimpose them, you also see this difference in decay between the wild type and the heterozygote. And this is statistically significant. And well, these are average traces, but this, of course, is in decay across all the cells I looked at. And I also looked in a cell homozygotes. In this case, I looked in layer 6. But what I did see is similarly a difference in decay. These are average representative cells. And then this is the difference across all the cells that I looked at. I also saw a small decrease in amplitude of events, and I saw a decrease in frequency, an increase in an inter-event interval, which kind of matches the decreased frequency of events seen in the GAT1 knockout in miniature iPSCs. It's a little different. This is spontaneous. That was miniature, but it does match. So given this data, I wanted to go back to my model where I was posing the questions that I spoke of earlier. So we know that there is decreased GAT1 expression and GABA uptake. based on biochemistry experiments and radioactive GABA experiments, radioactive GABA uptake experiments. And so now, given some of these electrophysiology experiments, I think have pretty clearly shown that there's a prolonged decay at the synapse, both in the evoked events and in the spontaneous events. But I did not observe an increased tonic current. I think this could be more robustly investigated in the future, given how pronounced of a tonic current there is in the GAT1 knockout. And I did not observe an acute deficiency in releasable GABA. But I think as a next step, we could also take a more rigorous approach, possibly a much higher frequency of evoked events. And this is what some of my colleagues have been suggesting. Okay, so with this, I'd like to close. Thank you much for your time. I'd like to thank my lab members and thank Caddy and the many undergraduate students that work with us.